Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
To fully understand how cells work, scientists need to know how their moving parts relate to one another in space and time. However, because of their size and the amount of data involved, visualizing cellular structures in 3 dimensions has proven difficult. Now, in a trio of new studies, 2 teams of molecular scientists have aimed to make it easy for everyone to see inside cells. The researchers are also making their 3D data, published in separate studies in early October, freely available for anyone to explore in order to allow researchers around the globe to probe their own questions about how cellular form impacts function. FIB-SEM works by taking an SEM image of objects embedded in resin, then shaving a tiny sliver of the sample using the ion beam and taking another picture. By repeating this process over and over, scientists can stack all the images to create a 3D rendering. But the machines need to shut down after 3 to 5 days to recharge the ion beam, and when they start up again, the amount of material the beam shaves off and the resolution of the image aren't as accurate. To reduce the inaccuracy after each shutdown and achieve the clarity needed to map the insides of cells, Xu added more stable ion beam hardware that allowed a higher current, troubleshot the control of the beam, and sped up the imaging of the SEM. Those tweaks, detailed in one of his team's 2 October Nature papers, improved the 3D resolution to 4 nanometers from 8 nanometers, which often meant the difference between organelles looking like clear, detailed structures and fuzzy clouds. For 2 years, 2 people worked full-time to manually identify organelles and outline their boundaries in 3D images from FIB-SEM. Heinrich says that the network uses the manually annotated images to learn rules, trying to adjust them in a way that the output it produces is the same as what the humans did. The network doesn't just look at each pixel and make a call about whether it's part of an organelle, it examines the pics. The scientists estimate it would have taken one person 60 years to manually identify the same number of organelles the algorithms can map in a few hours. The discrepancy in identification is often related to how abundant the organelles are in the human-labeled training sets. Heinrich says that more training sets and algorithms will help improve the mapping accuracy for all organelles and structures present inside cells. Adding the 4th dimension in the 3rd study, published in PNAS, a separate research group used computational approaches to infer the 3D structure of chromosomes. These 2D data would then be used to build 3D models, but this required a lot of assumptions about what would happen based on the DNA sequence and proteins that hold loops of DNA together. The researchers behind the new study didn't want to make that many assumptions. All we wanted to do is simply make a structure that follows the rules from experiments, says the study's first author Anna Lappala, a polymer physicist at Massachusetts General Hospital. When 2 X chromosomes are present in a cell, most of the genes on one X are deactivated through XCI to prevent developmental abnormalities. This silencing is initiated when 1X expresses a non-coding RNA called ZIST that coats the chromosome. The results of the structural modeling show that as the X chromosome undergoes XCI, it forms a dense core with a looser surface. Several genes known to escape XCI are located at the surface, which the researchers suggest might allow better access for the molecular machinery that expresses genes. What's really groundbreaking about this paper is that we figured out a way to visualize the 3D structure of the X chromosome based on a decidedly 2-dimensional map that one gets from high-C datasets, says Jeannie Lee, a molecular biologist at Harvard Medical School and co-author on the paper. The authors were also able to track the spread of cyst RNA on the chromosome over time, which they show in a video. Computing the future of biology. What I think is most significant about this kind of work, in the PNAS paper, is the attempt to explain something very complex, such as the structure of the genome and especially dynamic processes in the genome, based on the fundamental first principles of physics. Across all three papers, an important point to make is how critical computational analysis and modeling is for this area, adds Murphy. In the three new papers, powerful computers allowed the scientists to take huge experimental datasets and learn something new about organelles, chromosomes, and other structures without years and years of manual work. To speed up the analysis of data and progression of 3D cellular biology research even more, the authors of all the papers are committed to open access to data. It's much better to let the world see what we have invested in, Xu says. Xu has patented the new microscope technology, but it is free for universities and nonprofits to use, and the atlas of 3D cell data is freely available to explore. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.